Can you briefly explain your role in the lab?
Okay, I work in an analytical laboratory for years. Now I'm leader of physicochemical characterization team. My responsibility goes to the characterization of new synthetic compounds and support of preclinical studies, host method preparation, method establishment, development process for analytical methods for new substances, and also my team is responsible for in process control of manufacturing of new drug substances for preclinical and clinical trials. And for years, I was also responsible for ADME analytics. It means that all assays combined where we are using HPLC and mass spectrometers is I'm also familiar with this. So generally, I have quite broad knowledge and experience with different technique of liquid chromatography.
Yeah, can you talk to me a little bit about your experience with liquid chromatography?
Okay, generally in my lab we are working on different vendors. Most of systems which we are using, this is UHPLC, UPC systems, but we are also using HPLC, classical HPLC systems. We have different detectors, UVS standard, but Chronocat, light scattering. Also we have ion chromatograph, different mass spectrometers combined with HPLC systems. I also very familiar with 2DLC systems. We have very nice instrument from the Agilent. What can I say more? Generally, briefly, something like this.
So is the purpose of what you're doing analytical development, or is it also process development?
This is also method development, supporting of process development, because I'm not only an analytical chemist, but also I'm a synthetic chemist.
Mm-hmm.
My university, my PhD thesis was connected with organic synthesis. And because of this, I have knowledge in both of this area.
So can you tell me about the HPLC systems that you're using? Can you tell me about the setup that you have?
Okay, most of systems comes from Agilent.
Mm-hmm.
But we are also working on water system, Dionex Thermo. But for few years, I also work on Shimadzu. It means that I have general knowledge and possibility. I can compare this different kind of HPLC systems.
What do you like and not like about your current setup with, you've got Agilent Waters and Thermo, right?
Yeah. Generally, any of these vendors have their own, how to say, uh, strange and some major, how to say, many, they have pros and cons,
Mm-hmm.
different pros and cons. And dependent on this, what we want to analyze, or what expectation we have. If you would like to make repeatable analysis for process control, then we are using in most of cases HPLC or simple UHPLC from Agilent or from Waters. But when we would like to analyze compounds with cut detection, then we are using Dionex system. Yeah. So it depends of our needs. But generally, most of systems I have from Agilent, because of possibility of flexible configurations. Especially during method development, they have great scouting systems where we can connect different mobile phases, a lot of columns. So because of this is very useful for the analytic. I didn't need to establish method for different systems. I have one system when quite easy it is possible to switch from the condition A to condition B, C, D, and etc. So because of this, we decide to cooperate very strongly with Agilent. Waters, great system, especially I-class, UPC, UPLC, very repeatable, stable, very robust system. So it works for years without changing of the power. So it is great system.
And what about thermo? When is that useful?
Thermo. This is generally I work on different thermo systems. In our lab, for years, more than 20 years, I touch different thermo systems. I started from Ultimate 3000, and in my lab I have two Ultimate 3000. Generally, it's comparable in something in Agilent, but generally especially the sampler system is not such comfortable to work like in Agilent. They have the main disadvantage. This is a problem with washing of the injection system. It means that the carryover on Ultimate 3000 is much higher than on Dionex or than on Agilent or on Waters. It is non-comfort. The work on the thermo is not comfortable for analysts, especially if we have different kind of compounds. For example, in Agilent system, we have multi-wash system for sampler, which is very suitable and very, very, very flexible for us because we can dependently on kind of compounds. If we have hydrophilic, hydrophobic, it is not the problem to quite easy prepare the suitable combination of washing solvents. On Dionex, we have only one bottle, and we also keep in mind that, okay, if we wash the needle, we have to think that we are the same solution is using for the syringe. It means that it is second limitation of the Dionex system. Amount of volume of injection. Of course, the system in specification, it looks that it can we can realize quite broad volumes of injections, but in reality, repeatable injections we have above the 3 microliters. It means that it is some limitation, yeah. Especially if we have organics in syringe system, in washing system. This is the main limitation why we don't like working on Dionex. On Dionex, generally we have well-developed method. Yeah, this is good for this. Especially if we have one kind of analytes, for example, some polar analytes. Okay, this system is established and configured for such kind of analyte, and it works. But when we would like to switch for another, we have to change too much things.
So it doesn't have a separate wash bottle and a needle wash?
No, it is only one bottle for the syringe and for washing of the needle.
Okay.
This is the main disadvantage of this system.
So how many total HPLC setups do you have in your lab?
In this moment, 15. Yeah, 15. Yeah. Recently, I buy some new systems, yeah, 15.
How much of your work is just pure liquid chromatography, and how much of it is liquid chromatography coupled with mass spectrometry?
Generally, it depends because it depends of kind of work what we are doing. But I will say that about 60% for HPLC with UV detection, and 40% of work we spend on the mass spec.
And how do you store your HPLC systems? Are they on standard benches, or are they on carts or specialized benches?
Not standard benches, but it depends. Because our needs and our work is quite another than in quality control laboratories. We are working on research and development department. And because of this, some of systems have possibility to move from one to another, for example, mass spectrometer. So very important for us, it's also combination and quite easy reconfiguration of the software when we move the HPLC from one system to another. And most of instruments I have on benches, but for instruments for HPLC, I have on movable benches just to switch between different mass spectrometers.
And how tall do your HPLC stacks get?
Generally in benches okay, please repeat because I fix on some another thing. Please repeat the question.
How tall do your HPLC stacks get?
Generally it depends on the analyst, but some systems are some of this is quite tall. Especially if we have more more than standard amount of modules.
Mm-hmm.
But generally, everything is how to say is adopted for the analyst, because in laboratory I have tall different analysts, girls and guys. So because of this, for third person, it's not the problem. Yeah. But for example, for some girls, we have to use some stairs. Yeah.
Yeah, that's what I was gonna ask. So like I'm like 1.6 meters. Like would I have to use a step stool to get up to the solvent bottles?
Yeah. So for few systems, we have to use ladders, yeah, or some laboratory stairs.
Mm-hmm. And regarding the solvent bottles, like how many solvent bottles are you typically using in your setups?
Oh, it depends on our work. In some of this, generally in HPLC, we have 2 or 4 system for solvents for mobile phases. It depends if we have binary pump. In most of cases, we are using one and two clusters. So because of this, four bottles for the solvents, and some another for washing pump, washing sampler. It means that 6, 7 bottles we have on the top of the HPLC.
Mm-hmm.
Yeah.
And
But
that's
in
for
but
binary pumps.
it is on binary. But if we have, for example, a scouting system, it is quite difficult because on a systems we have 8, 11, 12 bottles.
Oh goodness.
Yeah, because we have solvent with switching valve. And for example, for the canal A, I can install more than 8 different mobile phases.
Mm-hmm.
And on B, C, and D, I can do that same or single bottles plus washing solvents. It
Mm-hmm.
means that 11, 12 bottles, it is some limitation. And if we have to do scouting, then we have to adopt everything. And we are using special bottles, quite narrow bottles,
Mm-hmm.
1 liter bottle, but quite narrow to arrange everything at on the top of HPLC.
Yeah, I was about to say, do they all fit on the top of the HPLC?
Yeah, it is possible. Yeah, we can fix it, and it is not the problem. But this is not the classical bottles as we have, but quite narrow.
Mm-hmm.
The Merck and Duran have such kind of bottles. So in Agilent, for years, they to each system they added such kind of bottles. So this is very useful.
Who prepares those bottles?
Once again, please, could you repeat?
Who prepares those bottles? Like who makes up the solvent bottles?
Okay. Generally, all mobile phases was prepared by analysts. We
Okay.
didn't buy commercial phases.
Mm-hmm.
In most of cases, because of limited stability of additives for the mobiles.
Yeah.
It's moreover, it's more cheaper.
Yeah. How often do you have to change or handle the bottles? How often does somebody have to like get up there and change
It
something?
depends on the stability of the mobile phases.
Hmm.
Uh, but we in our work we try to expand the stability, and because of this, we are using special cups. We filter the phases, and to avoid day by day removing the mobile phase, we are using addition of for the water phases. We are using addition of some organic to stabilize to stabilize water phase. Not more than 5%, but it is enough to keep the mobile phase for 1 week. But in some of cases, we have to exchange the solvents for 2-3 years, for 2-3 days.
Do you have any challenges with your solvent bottles other than having to try to fit them on the top of the machine?
No, it is not it's not the challenge. I will say some challenge we have on ion chromatograph, where we have to fix some special filters to avoid contamination by carbon dioxide from the air. Because sensitivity of the detectors used for the used in ion chromatography. But generally in classical UV detection, it is not the problem.
Mhm. All right, I want to show you these product concepts and see what you think. I'm pretty interested based on what you've said already.
Mm-hmm.
So the first one, can you see my screen yet?
Yes.
Okay.
Oh, it looks like Agilent.
Yeah, the first one is just a completely traditional HPLC. This is what I imagine your current setup probably looks like with the solvent bottles on top.
Yeah.
What are your impressions of this product concept?
It looks nice. Probably it looks like classic HPLC system. Is very fun, very similar for the newest instruments from Agilent.
Hmm.
Yeah, the concept is quite nice.
In
Only
hmm,
one thing. Um, uh, Legs of these systems
hmm.
are are too wide.
Tell me
Uh,
about that. Like
yeah,
the little
yeah,
feet down here?
yeah. And if you would like to combine few systems, uh, few systems side by side, it will
Then
be
they run into each other?
yeah, it could be problematic, but
Okay.
generally, it depends if you need to save the space in a laboratory. Yeah,
Hmm.
but generally it's okay.
Okay. So yeah, you're worried that like if you wanted to have like three of these side by side that these little feet would run into each other?
Yeah. It will be much more easier to clean to clean clean the space below the system. But generally, it's okay.
Hmm. So in a setup like this, like a traditional setup, how important is it to you that you have the ability to change the solvent bottle sizes, quantities, and types?
Generally, it looks okay. But as I said, uh, exchanging the solvents and bottles is not the problem. Yeah.
Mm-hmm.
If
But you like that do you ever, like, what size bottles do you tend to use?
no, generally this configuration is quite good for us. So
Mm-hmm.
we like this. So yeah.
Do you ever use bigger than 1 liter bottles?
From time to time, yes. If we have classical HPLC system and we have higher flow rate, then we have to use much bigger bottles.
Mm-hmm. Do you put them on top or do you put them to the
Yeah.
side?
In most of cases on the top,
Mm-hmm.
because of hydrodynamic of the pump, but generally, yeah.
What do you see as the advantages and disadvantages to this traditional setup?
I see generally, I see only advantages of such configuration. Um, Quite easy. We can combine a lot of systems in one place. So generally, it is okay. Classical HPLC system. Yeah.
Mm-hmm. Let me show you the next one. Um, so in this, the solvent bottles are, these are proprietary bottles that quickly
Mm-hmm.
connect without tubing. So there's no tubing in this.
Mm-hmm.
And they're below eye level and transparent.
Mm-hmm.
What are your impressions here?
Ah, interesting. Interesting. I will say that it will be suitable for person to see the amount of solvent. It could be okay. Uh, Quick connect. Interesting. If it will be work. One limitation, and I know that it will be problematic. Uh, it is good system if we have buffers with acids, not classical buffers as, for example, as a salts. When we are using salts for the for as a mobile phase, as a additive for mobile phase, very easy it is to crystallize uh, at the connection. And day by day, I'm sure that some drops from the phases will be contaminate this quick connect. And because of this, uh, in my opinion, it will be main disadvantage of this system. It looks nice,
Mm-hmm.
but in normal day by day work, it could be problematic.
So you're concerned that like a high salt concentration buffer would crystallize at this like
Yeah,
gasket?
exactly, exactly.
So what are your thoughts about the quick connection feature otherwise? Like, do you like it? Do you like not having to deal with tubing, or what are your thoughts about it in general?
General classic filter, as we have in when we put the uh, tube into the bottle, it's okay. It's not the problem. Yeah.
Mhm.
But this system could be much more problematic in normal operation.
Mhm. So you see more disadvantages to advantages
Yeah.
with the quick connect feature?
Yeah. And believe me, in normal work in quality control, when the people are not familiar with maintenance of the system, it could be problematic because, as I know, when the people switching in some instrument, then don't care about the cleaning of Quick Connect.
Mm-hmm.
It will be problematic. It looks nice, but it could be problematic in normal work.
Does the reduced number of solvent containers and sizes, would that be a significant limitation that there's only 5 of
Yes,
them?
second thing, it is the question is about material, because I don't think that it could be glass.
It is glass.
It is glass.
Yeah.
Oh, okay. Interesting. Okay. If it
But
is
does the number, there's only 5 containers here. Is that a
five
problem for
containers,
you?
okay, it is some limitation. In normal, in routine analysis, it could be enough.
Mm-hmm.
But if you would like to, it is not enough for development.
Mm-hmm.
But
So
for
this
routine
is
quality
enough for
control,
routine
yeah,
analysis, but not
yeah,
for method
yeah,
development.
yeah.
What do you see as the advantages and disadvantages of this setup?
Oh, main advantage that we don't have battles on the top.
Mm-hmm.
The disadvantage, uh, possible contamination of Quick Connect.
Mm-hmm. So how does this compare to
And
the
amount of possible bottles, yeah.
Mhm. How does this compare to the traditional HPLC that I showed you?
Uh, I prefer this traditional, but it could be, the system is interesting, but analysts have to very carefully work on this system.
Hmm.
Very carefully.
How would you feel about the system if the Quick Connect feature were removed and there were standard bottles but on the bottom?
Huh? It could be, it could be work. It could be work. Yeah. But if the pump will be on this place, yeah, it could be work.
Let me show you the next one. Um, in this one, um, this setup, the solvent bottles are at eye level and they're inside the device. They're the standard bottles that you would normally use, interchangeable in size. However, you'd only be able to use a limited number of bottles, probably up to 5 1-liter bottles. You could also probably use a couple 2-liter bottles, like 2 2-liter bottles and 1 1-liter bottle. Um, what are your impressions of this product concept?
Interesting configuration. Uh, Interesting configuration. It looks nice. Main limitation, amount of bottles. Not suitable for the scouting system, but very nice configuration for classical routine HPLC system.
Mm-hmm.
Main advantage that analysts will be see what happens in a bottle. They don't need to move the bottles on ice, because they will see. Yeah, when they move the drawer, it could be, yeah. It is a very nice configuration.
Yeah, how important is it to you that you would avoid handling bottles above eye level in a device like this?
Generally, it is not the problem. It's not the problem. This is problematic only in the case if you are using more than 1 liter bottle. If you are using 2 liter bottles, 5 liter bottles, but it is not happens too often.
Hmm.
From time to time, yeah.
Does the reduced number of bottles that you can only have like 5 1-liter bottles here, is that a significant limitation for you?
Uh, in classical routing system, yes, but it could be problematic if you will have some extra bottles for the washing of the sampler and pump. Because if we have binary pump with 2 canals, it means that we have 4 bottles.
Mm-hmm.
One additional bottle for seal washing of the pump,
Mm-hmm.
and one bottle for the for sampler washing. So it means that five that amount places for I expect more than 5 places for a bottle. 6 it'd be optimal.
Right, so it would end up being like that thermo device you talked about where it was using the same
Yeah,
wash
yeah,
and you don't like that.
yeah, yeah. But if you will have some special sampler wash, like in Agilent that you have multi-wash, you have 3 different bottles for solvent. Yeah. Of course, we can arrange this by changing the volume of the bottles. Uh, Here we have place for 5 1-liter bottles. If we will have half of liter, it probably we can put some another. But it depends on on target place. If you think about quality control laboratory, on routine laboratory, it will be okay.
So forgive me because my knowledge of HPLC isn't as deep as I wish it was. Is the reason you want a different wash for your seal and your needle because you want it to be a different solution or because of the volume of the wash?
Different solutions.
Okay, so you
Uh,
want it to be a
yeah,
different
yeah, yeah.
reagent.
Let's say it depends. In most of cases, in research and development, we are working on different kind of compounds with different properties. It means that solubility of these substances are quite broad, and
Right.
most of compounds have very low solubility. Because of this, we have to keep in mind problems with carryover.
Right.
Second thing, we have to, because of this, we are using multi-wash systems. It can give us possibility to efficiently wash injection system without carryover, especially if we have different kind of analyte. So because of this, I prefer multi-wash.
Right. So
Yeah.
you
And
want a different solution.
also, for example, water systems have
Mhm.
also 2 different bottles for the washing. It's
Right. It's a
very,
different reagent for
yeah,
the
yeah,
seal and the
yeah.
needle.
It is very useful.
Okay. Um, so how does this compare to the other two setups that I showed you?
Mm-hmm. If I could rank this 3 concept, uh, this first one will be the first. This one is very interesting. This one is very interesting as a second.
hmm
Very interesting configuration.
And then the one with the proprietary bottles is last.
The last, it will be the second as you
Yeah,
showed.
this is last.
Yeah, yeah,
Okay.
yeah.
All right. I want you to rank some things in order of importance. So, a 1 is not important to you at all, and a 10 is the most important to you. Does that make sense?
Mm-hmm.
How important to you is to have a flexibility of solvent bottle sizes, like being able to have a 500 ml bottle or a 2-liter bottle?
Generally, it is not important.
Okay, so like what would you rank that on a 1 to 10 scale?
6,
Uh,
5, 6, something like this. In medium. It's not,
it's a medium, so like a 5. You're
yeah,
neutral.
5, yeah, neutral, yeah.
Okay. How important is it to you to avoid handling or like to have bottles below eye level?
7, but it happens from time to time.
So it's pretty important to you, 10 being like the most important.
7.
Okay. And how important is that quick connection feature?
Ah, not important.
Okay, so like a 3 or a 2 or a 1.
2, 1 or 2,
Okay,
something.
a 1. Okay. Um, what's the optimal number of bottles that you would ever use? Like if you were building a system, how many bottles? Like what's the regular that you would use and what's the max number of bottles you would handle?
In regular applications, we are using 7 bottles.
7?
7.
And
In
then what's the
regular
most you
application.
would ever
6
use?
or 7. It depends on the instrument. 6 or
Okay.
7. It is classical amount of bottles on the top.
And what's the most you would ever use?
Ah, 15? Something or something like this.
11? Is that what
11, 12, as I remember, one case was when we have two solvent switch valves, and we try to install 15, 16 bottles, something like this. But
goodness gracious.
then we have to limit volume.
Yeah, you have to go down to smaller
Yeah,
bottle
yeah,
sizes.
yeah.
It would be great if they had square bottles then too. I feel like that would.
Yeah. But for scouting system, we are using UHPLC system when half of a liter is enough for the screening. So it was good. It was good for this idea, but in most of cases, 11, 12 bottles in a scouting is something typical. This is typical on this system.
So if you were the product designer and you had these concepts in front of you, are there specific features you would combine into a single ideal setup? Like, is there a system that would make the most sense for you, or is the traditional setup like ideal for you already? Or is there a way that you would change it based on what you've seen here today?
Uh, Generally, for routine work, quiet compact system, it will be something good. It's quite easy to maintenance, quite easy to work. Yeah, it will be okay. This system where the bottles will be in front of the user, not on the top, but in front on the side of the instrument of the it looks generally nice. Yeah, this one.
This one?
Yeah, this one. It looks nice.
For routine work, like
For
for,
routine work, for quality control work, it will be nice.
mhm. Is
It
there
will be
anything you
nice.
would change about it?
Uh, Generally, to change in the system,
Mm-hmm.
no, it generally it looks nice. The system could be probably quite wide, but
You're
not
worried
tall.
about the footprint?
Yeah. I think about the footprint. Yeah, but generally, it's not the problem. Yeah.
So this would be like your ideal system for routine work, but
Yeah,
for more method development sort of work, you would keep up with
standard configuration.
the standard configuration.
Yeah, absolutely yes.
So, is there anything you would tell the product designers that are working on this project that you would like them to know about developing a new HPLC system?
Space in thermostat and what's important, uh, even if we have routine work, in most of cases we try to install two columns. It means that possibility to easy fix the column in the oven in a thermostat. It's in some vendors it is some changing some challenge. Not in any system, it's quite easy. In Agilent, yes, they have some special tips to install more than even 8 columns. It is quite easy. But on Dionex, it is horrible work to
You can
install.
only put one column in the oven at a time?
Yeah. If you have one, okay. If you have two, okay. No. It is not. Sometimes it's problematic
Mm-hmm.
to keep in place the capillars. In the newest system of thermo, especially in Vanquish system, in this thermostat, they have two separate, how to say, two separate, um, they have two separate chambers for the two columns. It means that this configuration is very interesting and give possibility to fix the capillary and columns in one place and everything is okay. But it doesn't work on water system on Shimadzu now. This is important. Second thing, place in sampler for vials. In Agilents on waters, it's very useful where we have not to, where we have plates, and this is very important, very useful for analysts. What can I say more? If somebody will think about the HPLC, they should think about the connections, about capillars, especially in any system we have to install columns. It
Mm-hmm.
means that such a product like a Viper on thermosystem is very useful for analysts. Agilent have quite nice system, but from time to time, and especially if we have to change column for the waters or from waters to some another vendor, we have to change the fridge at the end of
Okay.
the mounting. It means that it is not. Sometimes it's problematic. Yeah,
So you want cross
because
compatibility?
yes, yeah, because analysts have to think about this that he have to remember that okay, I have column from waters and then I have to change the fridge to install the new one comes from either Phenomenex or Agilent or some another.
Mhm.
It is problematic.
That makes sense. Yeah, it would be annoying to have to do
Problematic.
that.
Viper's system for capillary is quite good, but the most problematic is also installing the waters columns. It can be possible, but uh, but analysts have have made some experience how to install.
Right. It's more difficult for
Yeah,
them to change
yeah,
the
yeah.
frit and
It's
put it
very
in.
difficult. Yeah, it is very difficult, but it works. It works.
So it's one of the best column systems, but
Yeah, yeah.
all right, this has been really lovely and insightful. I really appreciate your time today, Damien.
Thank you very much.
Thank you so much. I hope you have a great afternoon.
Okay, thank you. And
All
have
right. Have a great day.
okay, thank you. Bye.
Bye.